Okay, we'll get started now. So I'm Jason Ernst. I'm faculty here at UCLA. And I'm going to talk today about computational approaches for interpreting the non-coding human genome. So this is a small fraction, a portion of the human genome. And it represents, this diagram shows that the vast majority, about 99% of the genome is not coding for proteins. And that's relatively poorly understood compared to the protein coding regions. And there's a lot of interest in understanding these regions of the genome. One major motivator is these disease mapping studies, which have, in an unbiased way, found that the large majority of disease-associated variation for common diseases are falling into these regions. And many are likely having regulatory effects. So those of you who were at the retreat heard me talk about two different types of strategies that my group has been working on, one based on comparative genomics and the other based on high-throughput functional testing, such as massively parallel reporter assays. I'm not going to talk about those today, but you can watch the video if you missed the retreat talk. And today I'm going to talk about work we've done on epigenome-based strategies for better annotating and understanding the genome. So in this talk, I'm first going to give some general background and a review of some other types of approaches for epigenome-based annotation, and then talk about sort of some background into chromatin state modeling, then talk about epigenome imputation, and if there's time, some recent work on modeling temporal chromatin data, spatio-temporal aspects of it. So the genome is wrapped around, the DNA is wrapped around nucleosome proteins, which are comprised of an octamer of 8 histones. And these histones, proteins can be subject to different types of chemical modifications. And there's a lot of different histone modifications, and there's specificity in terms of which specific histone proteins being modified, which specific amino acid residue of the protein, what type of chemical modification, whether it's a methylation or acetylation, and how many occurrences of the modification, whether you have 1, 2, 3 occurrences. So you'll see notation like this for histone 3, lysine 4 is monomethylated, and here it it's trimethylated, and other modifications like this means histone 3, lysine 36 is trimethylated. And each of these have sort of a rough correspondence to what they might mark. But as I'll go through the talk, we can get more information about the genome by analyzing these modifications. And I'll show you how they're actually mapped. In addition to histone modifications, there's also DNA methylation directly on the DNA, as well as DNA accessible regions. The focus of my talk will be on the histone modifications where we have the greatest specificity. So what has driven a lot of the work in this field is this assay ChIP-seq, chromatin immunoprecipitation followed by massively parallel sequencing. So the idea here is that you have different antibodies that can recognize different modified forms of histones. So you can think of it as a lock fitting into a key. And the specific antibody is targeted to a specific histone modification. And what happens experimentally is you're sort of freezing the DNA with its specific modified histones, and then you're cross-linking and shearing the DNA, and you're extracting out specifically those locations where you have that modified histone modification, and you extract out that DNA sequence, and you're sequencing it, and then you can map it back to the reference genome. So what you get is density maps like this, showing you the relative level of different histone modifications in a population of cells in a given cell type. And this is showing you tracks for different histone modifications in the same cell type. And you're going to have different maps depending on which histone modification you map, as well as in what cell type it's being mapped in. And single experiment can give you on the order of 20 million reads on the location of modifications across the genome. So one of the sort of first things one can do with this type of data to annotate different locations in the genome is call peaks or domains of different modifications. And there's a lot of different methods to do this. As part of the roadmap epigenomics effort, which I'll talk about later, these are 6 different histone modifications. And there was peaks called using the MAX software. So what you're seeing here are the signal tracks for 6 different modifications, and right under it, these binary calls of where there's an enriched signal level relative to a certain background model. And then a lot of analyses, what people do is they'll take one histone modification and then do analysis based on locations of that peak. But this has certain limitations in that if you're interested in understanding what's going on at some specific location, if you just focus on one specific histone modification, you're throwing away a lot of information about what's happening in the epigenome at some specific location. So there's been a lot of effort into methods that can somehow reason jointly about multiple modifications at the same time. And one sort of class of this type of work is based on supervised methods. So this was a paper in 2007 by Bing-Ren's group, which made the point that there was this distinct set of chromatin signatures between promoters and what they were calling enhancers. So enhancers tend to be regulatory elements that are away from the start of genes. And what they were doing was they were taking annotated transcription start sites to represent promoters and locations of a specific protein binding P300 to represent enhancers. And what they showed was there was different signatures associated with different histone modifications, depending if it was in this class of promoters or enhancers. And then they developed one type of sort of simple approach to classify locations into promoters and enhancers. And since then, there's been quite a number of other methods using various supervised learning methods, such as random forest, neural networks, or supervised hidden Markov models. And I mean, these approaches can be useful, but there's limitations in that when you do the supervised learning, you're only sort of discovering explicitly what you're sort of training on. And in some cases with promoters and enhancers, it's not just you have this simple binary class of promoters and enhancers. There can be different subtypes. And then there can be other things in the epigenome that you might not even be thinking about. So this doesn't give you, allow you to discover things you weren't explicitly thinking about before when you do the supervised learning approach. Another strategy, and this was also by Bing-Ren's group with Wei Wang, was to do unsupervised based learning where they focused on identifying patterns in the epigenome based on clustering fixed window lengths. So in this case, they were clustering all the promoters based on a set of histone modifications for fixed windows. But this, I mean, it's useful, but has the limitation that these approaches, not everything fits into one sort of uniform fixed window length, and a lot of locations aren't getting assigned to a specific cluster. So this sort of motivates another strategy for annotating the genome or epigenome in an unsupervised way based on chromatin state modeling. So to give some intuition here, on top of the DNA sequence, we could have these different histone modifications represented by these different shapes. And what you might notice is we could have reoccurring combinations of marks, or we could have broad domains of marks. And what we would like to do is have one model which would de novo recognize these patterns. And the assumption would be the reason we're seeing these patterns reoccur in the, across the genome is that they're likely signifying some class of elements in the genome. So this is another view of what we're trying to do here. We're taking these different tracks of epigenomic maps, and we're trying to reduce them to a single state assignment at each location. And we're going to learn these patterns de novo. So the modeling approach that I had developed was based on a multivariate hidden Markov model, and we call the method ChromHMM. And the idea here is that we're going to pre-process the genome into 200 base pair non-overlapping intervals. And then for each mark in each 200 base pair window, we're going to have a binary presence or absence call if the number of reads for that mark exceeds what we would expect based on a Poisson distribution or at a specific significance level. And then we would model the mission distributions with the product of independent Bernoulli random variables. So depending on which state we're in, we have a different probability of observing each modification. I'm showing you here marks with a high probability in these different states. And then we also have underlying this transition probabilities that capture the likelihood of transitioning to different states or staying in the same state. So this, in principle, could allow us to differentiate a location like this and a location like this, where both locations we might not have observed any signal for a specific mark, but we could reason about the neighborhood information to infer here we might still be in this specific domain, while here we're in this other state. And all these probabilities are going to be learned de novo from the data. And one of the modeling choices here we're making is this focus on these binary presence or absence calls. And that forces the model to focus on modeling unique combinations of marks. So this was an application to a set of data in CD4 T cells by Keiji Zhao's lab. And what you're seeing here, the rows correspond to different chromatin states, the columns to different modifications or marks. And what I'm giving you here is an example. Now that once we learn one of these models, we would go across the genome and assign each location to an instance of one of these chromatin states. And then we can start comparing these annotations to other types of annotations. So what I'm showing you here now is enrichment of these states for locations of zinc finger genes and CAP1 binding. And what this is showing here is there's an over 100-fold enrichment for zinc finger genes, specifically in this state, which has this unique combination of the presence of the histomodification H3K36ME3, H3K9ME3, and H4K20ME3. And it turns out that a large part of an abstract of this paper was describing specifically the presence of trimethylation of these three histomodifications, and the lack of these other marks were associated with CAP1 and zinc finger binding. So this is an example by applying the sort of appropriate computational machinery that we can discover de novo a very specific biological concept and annotate the genome systematically into instances of this concept. And then other states, we could find other patterns. So that, for example, we had one state which specifically enriched or peaked at the end of transcription and this wasn't something we would necessarily expect a priori to find. So once we had this model, we would go across the genome and compare each state to enrichments of, or to other annotations and compute various types of enrichments. And then this gave us different candidate annotations of the genome into different classes of promoters, different classes of transcribed regions, different types of active intergenic regions. So these are, can be associated with these distal regulatory elements, which can show signs of transcription factor binding and evolutionarily conserved elements, but can be far from the starts of genes and could be difficult to detect without this type of data. And also different types of large-scale repressed and repetitive elements as well. And I mean, one question that often comes up is how do we sort of focus on one specific number of states? And in practice, what we do is we learn models across different numbers of states, and then we see at what point we can still make sort of meaningful biological distinctions at the level of analysis we're interested in conducting. So it can be easier to set a lower bound where you know that you need at least a certain number of states to find biological distinctions of interest. It can be harder in some cases to set an upper bound. So that was all in one cell type. The power of this epigenome data compared to the genomic data is that it gives you annotations that can be specific to cell or tissue types. So if you move into different tissue types, quite often what you see is the location of different modifications can change, but generally the combinations of marks and the state definitions are relatively stable. It's just the locations that vary. So this was work in collaboration with Brad Bernstein's ENCODE production group, where we took a subset of modifications and they mapped them genome-wide in a subset of, or a set here of 9 cell types. And one of the questions is then, how do you go about analyzing this type of data where you have multiple marks mapped in multiple different cell types? And this is showing you all the data at one location. The different colors correspond to different cell types, and then each individual line is a different mark in that cell type. So what we did was we applied conceptually the same modeling approach, but treated different cell types as if they were effectively like different chromosomes. So we learned one common set of state definitions, but then we would have cell type-specific state assignments. So what you're seeing here is a chromatin state model here. The rows, again, correspond to different states, the columns different marks, and the darker blue means a higher probability of observing that mark in that state. So again, we can go across the genome, annotate locations to being instances of certain states, compute enrichments for other types of information, and then start giving interpretations of these states in terms of it's an active promoter, a weak or a poised promoter, strong or weak or poised enhancers, insulator regions, transcribed, weakly transcribed, polycomb repressed, heterochromatic, and repetitive elements. And this is another view of what we've effectively done here is we've taken raw input data like this, showing the same genomic loci in 4 different cell types, and we've reduced that into one chromatin state call at each location, each cell type. So now somebody can go into the genome browser and quickly scan across any genomic loci and get a sense of its chromatin state environment in each cell type, because we have the same set of chromatin state definitions in each location, but the cell type assignments can be dynamic. So one of the applications of this is for interpreting disease-associated variants. So this was, when we did this work at the time, there wasn't sort of too many genome-wide annotations available. So there were some, questions about sort of why all these non-coding GWAS hits were falling into these non-coding regions. So we started to revisit some of these GWAS hits, and this is giving an example for looking at erythrocyte phenotypes associated with hypertension and cardiovascular disease. And the columns correspond to different lead SNPs, and what you're seeing at different independent loci, and what you're seeing here is there was a block of these SNPs specifically in what we were characterizing as strong enhancers in K562 cell types. And if you're familiar with the biology of these different cell types, this is specifically the biologically relevant cell type. And we did this analysis just based on these lead SNPs, primarily from the NHGRI GWAS catalog at the time, and showed for a number of different studies that we saw this association for the GWAS SNPs with what we were characterizing as enhancers in the biologically relevant cell type. So immune-related GWASs were falling into the lymphoblastoid cell type, and some liver lipid-related traits were falling into HepG2, which is a liver carcinoma cell type. And since then, there's been quite a lot of work in this area at the intersection of the GWAS and epigenomic data using sort of far more sophisticated analysis approaches. So this question of how you sort of model multiple cell types, what we took here was an approach based on concatenating the different cell types, but it's useful to just sort of see with existing ChromHMM software, there's actually 3 immediate ways you can apply the software when you have data from multiple cell types. So one approach is to just treat the cell types as if they were independent and learn separate models for them But then you won't have the state assignments being directly comparable. The approach that we took that I just showed you, where you learn one common set of state definitions, but you can have cell type specific assignments. And another approach you can take is you can just stack all the data you have. And in this case, what you're getting is one annotation of the genome based on all the data. And you're not getting these cell type specific state assignments. And this approach we actually used in this paper here studying IPS reprogramming. So this is an example of a stacked model. And what it allowed us to do was sort of discover both sort of classes of promoters and enhancer states, but also annotate them by their trajectory across different stages of reprogramming process, where you go from, in this case, a skin cell to an embryonic stem cell. So I want to note that so there's quite a number of other methods that have come out for chromatin state modeling. So one method, actually, HMM-seg, which predates ChromHMM, was focused on just the problem of modeling two different states. It wasn't learning sort of these rich annotations of enhancers and promoters. Another method, which was developed around the same time as ChromHMM, Segway, there's a number of differences, but one sort of primary difference with Segway and some other more recent tools such as EpiC-Seg and GenoStan is the approach they take to modeling the mission distribution. So they use various continuous or discrete count distributions. And there's trade-offs involved in this, but one issue when you're modeling sort of more this continuous range of values is it can potentially be easier for the model to overfit low-level signal intensity or focus more of the modeling power on modeling different ranges of signal opposed to novel combinations, which sometimes can correspond to very unique biology, such as the zinc finger gene example or bivalent promoter states. And there's been other types of variants proposed, and all these, or most of these are referenced in a recent paper in Nature Protocols where we explain in a tutorial fashion the ChromHMM method and a description of some of the recent work in this area. There's other approaches that try to do specific extensions, one looking at hierarchical modeling, another approach trying to speed up the model learning, though it decreases the ability to distinguish different types of broader domains. One approach that does strand-specific annotations. And there's a number of different approaches that have been proposed to handle the situation where you have multiple different cell types and to sort of leverage that type of structure in either inferring the model parameters or the state assignments. But for the general use case, in most cases, the sort of standard ChromHMM method most people have been adopting for a variety of applications. So now I'd like to talk about another problem, and this is epigenome imputation. So in this approach, or it's motivated by some of the roadmap epigenomics data, which tried to map various different histone modifications in a large number of different cell and tissue types. And what you're seeing here is the rows correspond to different cell or tissue types and grouped into these major groups, and then the columns are individual histone modifications or other related chromatin marks. And blue meant that some mark was mapped experimentally in that corresponding cell or tissue type. And white meant there wasn't any data available. So in this project, we applied ChromHMM to these 127 samples that had these 5 histone modifications available in it, directly mapped experimentally. And what you're seeing now is one of these chromatin state annotations across these 127 samples into these 15 different chromatin states. And then we were also interested in including additional marks, so we included H3K27 acetylation, which can be associated with active promoters and enhancers. But one issue was we only had this data available in 98 cell and tissue types. So we learned this model just on those subset of samples. And we could continue adding marks and reducing the number of samples that we would have coverage of. But that wasn't particularly satisfying. So we decided to focus on a different approach here, which I'm going to tell you. And what we did was we first computationally imputed what one of these experiments would look like, leveraging the fact that we have other marks mapped in any specific cell type, and we've had that mark mapped in other samples. And I'll give more detail about how we did that. And then we inferred one of these chromatin state models now based on the 12 marks. We were most confident in their imputation based on having been mapped in enough other cell and tissue types, and provide an annotation of the human genome into 25 different chromatin states across all these different cell and tissue types. So how we actually go about going from a grid like this to a grid like this was based on using an ensemble of regression trees where we leveraged two classes of features. One is having other marks in the target cell type, and the other was having the target mark and specifically in similar cell types. And we're assuming we have no data available for the data set we're trying to predict here. And we're not only going to actually predict the data sets which we don't have any data available for, but we're also going to generate an imputation of the data sets which we do have data for. And it turns out that there's a number of benefits actually to the imputed data, even when you have the observed data available. I mean, one is you can compare against the observed data, and that can be useful for quality control. But it turns out that there's a statistical robustness that you gain when you combine information from lots of closely related experiments, opposed to just relying on the data from sort of one or some very small number of replicate experiments. So there's effectively a denoising process going on. So the approach that we did was we trained an ensemble of regression trees where we both used the data from the mark in the target cell type and that same mark in other cell and tissue types. And what we had was features based on those other marks in the neighborhood around the target and k-nearest neighbor-based features for our target mark in other cell types. And this was trained based on an ensemble of regression trees at a 25 base pair resolution, except for DNA methylation, which we did at 1 base pair resolution. So just a quick overview of the regression trees. I mean, it's similar to decision trees if you've seen them, where what you do is you have at each node a split feature where if it's less than some value, you go one way. If it's greater than some value, you go the other way. And then at the leaves, you have numerical values that you're predicting on. And part of learning, you're deciding what specific features and what the split values are. And it's an ensemble of these different regression trees where we're training on each of the cell types for which we have the mark available, and then applying it to the cell type for which we don't. So what you're seeing here is a visualization of the output of this method, where blue is the observed data and red is the imputed data, where we're not using the observed data to inform that prediction here. And this is now zooming in and showing that we can capture very fine resolution features as well. And we also quantitatively evaluated this. So in this case, we compared to two stringent baselines. One was the best case for using any single data set for that mark, no matter how well we would predict which cell types to use in place of the one we're trying to fill in. So often this is how people work in practice. If they're missing some mark in a specific cell type, they'll sort of guess what is the most relevant cell type that they have and use it in place of that. So no matter how well you make that selection, this imputed data had better correlation with the held out observed data. And another baseline would be just averaging that target mark in all the cell types for which we have the data available. And then another point I was making was we might actually have sort of benefits over using the directly observed data. And this is one approach to sort of argue this. What you're seeing here, each pair of heat maps corresponds to one specific mark. And the top row corresponds to the observed data, the one below it, the imputed data. And it's showing you a correlation grid where yellow means higher correlation between pairs of experiments. And these experiments are ordered based on the known biological grouping of the cell types. And white means there's no data available for that mark in that sample. And what you should be able to see visually is there's a much clearer block structure here than when you look directly at the observed data. Through the imputed data corresponding to the known biology. And we can quantify this based on, for example, the area under the ROC curve when we're trying to predict just based on the correlation between two experiments, if we can predict whether that pair of experiments would be annotated to the same major biological grouping or different ones. And this was, the most dramatic difference was for these broader marks, which tend to be more diffuse in their signal and harder to map and less cell type specific. Just to note that recently there was another method for this problem proposed by Bill Noble's group, and those of you who are at CGWI saw his talk, and they sort of took a completely different approach to this problem using a tensor-based decomposition. And it turns out this approach, based on the metrics we were using, wasn't actually better, but they show that it's complementary, that combining the two methods led to an even better prediction. And then in the remaining time, I'd like to talk about a new method that we've developed. This was led by my student, Pete, for modeling spatial-temporal dynamics. So one of the aspects of a lot of the epigenomic data that I've been mentioning is it's very dynamic. So oftentimes people study some biological process where they'll do a time series of these experiments. And a lot of these chromatin state modeling approaches can be applied to the time series data. But what will happen is you're sort of annotating each position in the genome, giving it some specific chromatin state call, but it's sort of missing one aspect of this data of this notion of domains expanding or contracting here. And there's sort of recognition that like the length of different modifications can have association with important biological other features. For those of you who've heard about, for example, super enhancers, those can be characterized by these long domains of marks. So what you're seeing here is an example of a region which, based on the signal level, looks like it's steady across time points. So if you look directly at peaks called independently, it might sort of miss this dynamic. While over here, if you sort of look at the signal, you can see that there's an expansion, at least at certain time points between here and here, and here. While in this location, it looks like there's a contraction of the signal. So we wanted to have a computational approach that would capture this type of modeling. So the approach that Pete developed can be sort of broken up into two phases. One is we find blocks of region for the modification we're trying to look at here. And then once we focus on specific blocks, then we classify each time point into a peak region and a background region within the block, and then an associated dynamic of whether it's a contraction between time points an expanding region or a steady region. So this is a more detailed view of what we're doing. Once we have one of these blocks at each time point, each position will be associated with either the peak or the background region. And then between time points, it's either in a steady where it has to have exactly the same position across time points, or an expansion where it can expand, or if contraction would get smaller, the coverage. And then associated with the background and the expansion, we can have different expectations of the signal. And Pete developed this probabilistic model that can be represented as a graphical model here, where we have different components of this. We have the signal, which is modeled with the negative binomial, which differs whether we're in a peak or a background region. Then we can also have different boundary dynamics, which can either be steady, contract, or expand, and we can have a prior multinomial on this. And then we can have also the B variable corresponds to the position we're in of the boundary of the peak region, and we can have between two time points probability distribution of where that boundary will be, and that depends on the boundary at the previous time point and the dynamic of whether it's an expand, contract, or steady region. And then there's some variables to tie it so that the boundary on the left is always before the boundary on the right. And all these model parameters can be learned with an expectation maximization algorithm. So this is an example of a location based on a T-cell development process in mouse, showing, in this case, the peaks being called independently by the Max software. We don't use those directly. We work off of the raw signal. And then what you're seeing here is the output of this Chrome method, where it's showing you the boundaries it infers, and then colors in red places that it associates with expansions between time points. And then you can see a similar thing for a location which has a lot of contractions going on. And one observation that came out of this type of modeling approach is that there was an asymmetry at promoter regions. So this is showing you a location of one gene across time points, and what you're seeing is the promoter signal for this histone modification H3K4ME2 expanding at multiple time points, while it held on the right side into transcription, but it held steady on this other boundary. While for this location, we saw this contraction across time points away from the transcription, and then this boundary held steady. And this was something we analyzed more systematically and found that this held for several histone modifications, that there was this asymmetry of expanding into the transcribed region or contracting away from it. So I'd like to summarize. I covered a variety of approaches people have been developed for annotating the genome based on epigenomic data. In particular, I focused on a chromatin state modeling approach, which both allows discovering de novo epigenome annotations or states and annotating the genome based on them. An approach for imputing the epigenomic data, which allows us to expand these annotations to cover more marks and cell types. And then a new approach for modeling the spatio-temporal dynamics of some of these chromatin marks. And I'd like to acknowledge the many people involved in a lot of this work, both from my group and some other collaborators. And thank you for your attention.